It is with great satisfaction that we are meeting virtually for the fourth workshop on innovation in biomedical engineering, an event organized by the undergraduate and graduate program in biomedical engineering at the Federal University of São Paulo. I would like to welcome all participants to the event. Professor Tati?
Yes. Hello. Olá, sejam bem-vindos à quarta edição do Workshop de Inovação em Engenharia Biomédica, hoje nosso segundo dia do evento. Esse evento foi organizado pelos cursos de graduação e programa de pós-graduação em Engenharia Biomédica da UNIFESP. Todos bem-vindos. Gostaríamos também de dar as boas-vindas à nossa palestrante. Fábio?
Well, for those who don't know me, I'm Professor Fábio Aoki, and I am a professor in biomedical engineering at UNIFESP. It is with great pleasure to introduce you to our distinguished speaker on session 5, Bioengineering, Dr. Alba Elena Marin Araujo. Is that correct? Well, Dr. Marina Araujo is a health specialist with over 15 years of multidisciplinary experience in healthcare, therapeutic options, bioengineering, and research and development. Dr. Marin holds a degree in medicine from the Universidade de Zulia, Venezuela. She also holds a master's degree in applied science and biomedical engineering from the University of Toronto, also called UFT. And also served as a postdoctoral research fellow at the University Health Network. By the time I was working with her at the same time, right? And in her postdoctoral research, she worked in tissue engineering, where she applied clinical knowledge and research and engineering tools to implement tissue-engineered tracheas for use in transplantation. She currently works as a clinical specialist at OTwo Medical Technologies in Canada. So right now, I'd like to give the word to Professor Tatiana.
Thank you, Fábio. Dr. Alba, thank you very much for having you here with us for our workshop. And now we are going to introduce yourself in Portuguese, and then give some instructions to our audience. So, excuse us. A doutora Alba Marin Araújo é especialista em saúde, com mais de 15 anos em experiência multidisciplinar em saúde, opções terapêuticas, bioengenharia e pesquisa em desenvolvimento. Doutora Alba é formada em medicina pela Universidade de Zulia, na Venezuela. Também tem mestrado em ciências aplicadas em engenharia biomédica pela University of Toronto. E também atuou como Research Fellow na University Health Network, também no Canadá. Em sua pesquisa de pós-doutorado, trabalhou em engenharia de tecidos, onde aplicou conhecimentos clínicos e ferramentas de engenharia de pesquisa para implementar traqueias para o uso em transplantes. Ela atualmente trabalha como especialista na clínica OTwo Medical Technologies, também no Canadá. Seja muito bem-vinda ao nosso evento. Nós gostaríamos, antes de iniciar a sessão, de passar algumas instruções à nossa audiência, né, ao público que está nos assistindo. Gostaríamos de avisar que todas as perguntas, elas podem ser enviadas nos comentários, no chat do YouTube. Vocês podem fazer as perguntas, apresentar as perguntas em português ou em inglês. As perguntas feitas em português serão traduzidas para a doutora Alba para o inglês e ela as responderá. E também gostaríamos de avisar que a lista de presença, vocês devem responder a lista de presença através de um link que estará disponível no chat ao longo da palestra. Desejamos a todos uma excelente sessão e eu passo agora a palavra ao Dr. Fábio, professor Fábio, para que possa ser iniciada a apresentação da doutora Alba. Muito obrigada.
Okay, so Dr. Alba, each speaker has around 60 minutes of presentation, followed by 30 minutes of questions. I would like to ask you to share the presentation on your screen now, and you can start your presentation. Okay, now you can turn on your microphone, and then the screen will be yours, okay?
Yeah, thank you, Fabio, and thank you, Tatiana, as well. I'm really excited about today, and as Tatiana and Fabio mentioned, I worked as a clinical specialist here in Canada in a company that it's called OTwo Medical Technologies. As they mentioned, I'm a medical doctor with a specialization in internal medicine and nephrology, and I worked as a physician back in Venezuela. Here in Canada, I pursued a master's degree in biomedical engineer, as they mentioned, and I dedicated my master's and my postdoc to tissue engineering and regenerative medicine. So now, as I said, I'm working in a company specialized in manufacturing mechanical ventilators and devices for patients' transports in pre-hospital settings. So as a master's student and then as a research fellow, I was part of University Health Network, as it was mentioned before, and also I was part of Atom's lab, which was part of the engineering department at U of T, and also I was part of IBBME at University of Toronto. So I was my projects, or the projects that I work on, were founded or are founded by Medicine by Design. The engineering and thoracic surgery departments have worked in collaboration for several years in projects related to tissue engineering, lungs, and tracheas. So let's begin our demonstration on our presentation today saying that this is a quote, I never thought before how strange the notion of a transplant list is. The only list I've ever really given thought to were groceries lists or to-do lists. I never thought there was such a thing as a list of names, people waiting for someone else to die. So there's no need to say that waiting for an organ is a big issue. So allogenic graft or allograft, that is tissue donated from another human being, are presently irreplaceable tissue grafts used to replace damaged tissues or non-functional organs to restore their normal function. So for decades, organ shortage has been the main disappointing barrier in transplantation medicine. Nevertheless, we have not yet found ways to overcome this complex issue of the shortest of organs for transplantation, that is kidney, pancreas, heart, lungs, and now we're going to focus on tracheas. So our patients around the world still die in acceptably high numbers in waiting lists. So organ and tissue bioengineering is a possible option to overcome this problem. One approach is based on organ decellularization and recellularization. There are several approaches like bioprinting or using prostheses. Like there are a lot of possible approaches to give a person solve the issue of replacement to a patient, but we're going to focus on decellularization. This is the process by which cells are removed from tissues or organs, but all the essential cues for cell preservation and homeostasis are retained in the three-dimensional structure of the organ and its extracellular matrix components. And we have, this would be the scaffold that we see on the slide. During tissue decellularization, maintenance of the native ultrastructure and composition of the extracellular matrix, we're going to call it ECM, is extremely acceptable. For recellularization, the scaffold, or that ECM, is seated with cells, the final goal being to form a practical organ. So the biological properties of the ECM that we have mentioned that remains when a variety of decellularization methods are used make the recellularization feasible within a bioreactor, this device that we see a schematic on the slide. And this would be a perfusion-based creating a artificial organs. So this recellularization would be using recipient stem cells ideally, and therefore immunosuppression in that case is no longer required, which is also another problem that we have. Those patients receiving or getting an organ from another person require immunosuppression that entails like a lot of complications in the future, in the short-term and long-term future. So in the future, like decades or maybe 5 to 10 years from now, recellularization procedures may solve the problem of organ assembly on demand. So the field of tissue engineering has made considerable strides since it was described in the late 1980s. So the advent and subsequent boom in stem cell biology, emergence of novel technologies for biomaterial development, and further understanding of developmental biology have contributed to this accelerated process. Tissue engineering studies have until now been able to partially increase cell engraftment by reinforcing cells and cell contact in animal models and by providing non-immunogenic matrices before transplantation. So this would overcome that issue, that state of immunosuppression. Therefore, that state of immunosuppression wouldn't be a problem anymore. So now let's focus on tissue engineering at trachea specifically. So tracheal injuries, stenosis, and malignancy demand surgical management or tracheal reconstruction. However, tracheal reconstruction or replacement of large segment fail due to the lack of a functioning epithelium, immune rejection, and also graft ischemia. So repair and replacement of tracheas have been evaluated and included assessment of autologous and non-viable tissues, foreign materials, as I mentioned before, prostheses, and naturally derived biological acellular scaffolds. And this is what we are going to focus on in this presentation. So for long segment tracheal defects, tissue engineered tracheal replacement have shown potential as a therapeutic alternative. So generation of biological tracheal replacement involve the repopulation of fully or partially decellularized tracheal scaffolds inside a bioreactor system before the in vivo implantation. So ideally, a bioreactor system for tissue engineering tracheas and any other organs, but as I said, we're going to focus on tracheas, should be designed overcoming limitations such as a lack of parameter control and monitoring, for example, temperature, flow rate, rotational speed, and also the bioreactor should provide controlled flow delivery to achieve a homogeneous hydrodynamic environment that would improve recellularization outcomes. So as you can see, and by now you will think that it would require not only a biologist or a chemist or a chemical engineer, but also other disciplines that will make possible the creation, the development of this engineered organ. So one approach, as mentioned before, of tracheal recellularization and recellularization focuses on specific tracheal components, as we can see on the slide. Complete removal of chondrocytes and repopulation of the tracheal chondroid matrix to achieve functional tracheal cartilage may have been unrealistic. So for more than 30 years, it has been stated by several authors that cartilage per se does not initiate immune response. So that is, the cartilage is immune privileged tissue. So our group at UHN has focused for several years on radically different paths for the past decade. So we try to develop a hybrid graft where the epithelium is removed and the immune privileged cartilage is preserved. Tissue engineering a trachea would entail different steps. The very first one would be a partial decellularization. This is our approach. And from now on, we are gonna call it De-Epithelialization. So we are gonna remove only the epithelium and we're gonna keep the cartilage. The trachea would be exposed to different detergents in this decellularization or partial decellularization, and then would seed epithelial cells onto the scaffold that we created, and the re-epithelization would be initiated. So this whole process would be held in a specialized bioreactor. So a bioreactor created just to do this process with tracheas. Our group developed a novel de-epithelization process which involves using different detergents and washing steps. And this short protocol, as we see on the slide, removes the tracheal epithelial layer, keeping most of the cartilage alive, and it would take the use of SDS and Triton as specific detergents and specific washes during a specific period just to get a de-epithelized trachea. So in 2019, we published this paper that we see here, De-Epithelialization of porcine tracheal allografts as an approach for tracheal tissue engineering. So this was the very first time that we accomplished a de-epithelialization. And this here for this project, long segment of pigs' tracheas were de-epithelized using our bioreactor. Then we assessed their chemical and physical properties. So remember, the ECM must be of such quality that epithelial cells would attach. It wouldn't make sense to have a bunch of detergents, the exposure of the trachea to a bunch of detergents, and then at the end of this decellularization or partial decellularization, the cells won't attach. So we wouldn't be accomplishing the whole goal of creating a new organ with the entire components needed to that organ to be functional. So we use the very first, one second. So we used the very first generation bioreactors. That was a bioreactor that was created a decade ago. And then also we developed the second generation bioreactor, but we're going to talk about that later. So its design, as I said, the generation bioreactor was modified along the way. So it met our needs, our specific needs. So here is a short 3D demonstration of how the bioreactor looked like right now. So we can see how the bioreactor is designed to let the trachea rotate inside. So also how the tracheal lumen and the other side of the trachea are independent. So let me start again the whole video, okay? So we have the bioreactor. The bioreactor would be assembled or set up inside a biosafety cabinet. Everything have to be in sterile conditions. And we have the body of the bioreactor. We also would have attached the trachea inside the bioreactor, and like a lot of components would have to be assembled in order for the bioreactor to work with a motor attached to the bioreactor. And then during the process of de-epithelialization and later on during the process of re-epithelization, the trachea will be rotating and the bioreactor would be completely sealed inside the incubator. So we're going to talk about this in detail during the presentation. Okay, so this is how the bioreactor look like. It's a rectangular box. Before it was made of plastic, but now we change and it's made of stainless steel. In order for it to be autoclavable and more resistant. So all different components, as I said before, must be autoclaved or disinfected before its use. So after harvesting the trachea at the operating room, we wash the trachea a few hours. After the de-epithelialization, before the de-epithelialization, to remove some bacteria and fungi, and these microorganisms are normally present on its lumen. After several washes for 3 hours, the bioreactor will be set up in sterile conditions in a biosafety cabinet, as I mentioned before, and after we will place the trachea, the washed trachea, and the bioreactor would be closed and sealed, and it would be moved to an incubator where the de-epithelialization and re-epithelization will be performed. The de-epithelialization would be first. It would take 5 to 6 hours if we take into account the setup of the bioreactor, and then we will re-epithelize the trachea. So on this slide on the left, we can see the double chamber bioreactor system configuration during the de-epithelialization. So this is the De-Ep circuit, and it's the tubing is in light blue. So the detergents will flow into the lumen using a pump, and media will be surrounding the trachea to nourish the cartilage. Okay, so these are the two chambers, the luminal chamber and the outer chamber of the trachea. So as I said, after that 5 hour de-epithelialization, we get the cartilage rings with 65 to 75% viability. After performing a live day staining, we can see on the right, in red, it's those cells that are not viable, those cells that were partially exposed to the detergents, and the viable cells are in green. And this is a cross section of several cartilage, and we can see that the immune privileged cartilage is still viable. So, as I mentioned, we can compare on top in label in A, we can compare the fully viable native ring, and that was obtained from a non-de-epithelized trachea. So those tracheas that were harvested, and then we assess the viability right away after harvesting in the operating room, and then we compare those findings with a de-epithelized trachea that we have labeled B. So as I mentioned, the green area corresponds to viable tissue, whereas the red areas correspond to non-viable cartilage. So as I mentioned before, after the de-epithelialization, we can start the re-epithelization. So for this process, we had to have a different system composition, and we have here a schematic representation of the recellularization bioreactor system, showing the addition of sample ports and filters, flow sensor, as we see in black, and temperature sensor to the same bioreactor system used for the de-epithelialization. So the body and the main components of the bioreactors are the same. The difference is the way that the circuits are placed, and the thing that we are not flowing and instilling detergents. We are using media, and in both chambers, in the luminal side and the other side. So for the very first experiments, we used previously published parameters. So we are talking now about the re-epithelization. So when I started working at the lab, Fabio had developed this de-epithelialization process, and as I mentioned, we published those results, and we wanted to move forward to the re-epithelization, use that de-epithelized trachea that we were able to get from Fabio's results, and then re-epithelize that trachea that we got. So as I said, for the very first experiment, so we have done different experiments for different projects in the past, but we didn't use for those the re-epithelization. We were aiming to have a totally decellularized trachea. So and we were not able to accomplish a fully re-epithelized trachea. So now we are focusing on the re-epithelization. So we use previously published parameters, for example, flow rate of 1.5 mils per minute and rotations per minute of 5 RPM. We also focused on Bs2Bs cells, which is a cell line commonly used in research for proximal airways. We compared two different cell concentrations for cell seating, so we didn't know for sure that the parameter that was used before was the ideal one, or if we can accomplish good results with half of the cell concentration that we were using before. So as I said, we use 500,000 cells per centimeter square versus 1 million cells per centimeter square. So we represented the number of cells found in different sections of the trachea and concluded that 1 million cells per centimeter square of trachea is ideal for trachea repopulation with Bs2Bs biperfusion cell seeding at 5 RPM using our bioreactor system. As we can see here, the high cell concentration is in green and the low cell concentration is in gray, and we focused on our following experiments on those of tracheas with high cell concentration seeding. Beside that, we measured during those experiments the metabolic cell activity in the luminal chamber using Presto Blue assay. In the graphic, we can compare that trachea seated with high cell concentration, that is 1 million cells per centimeter square of the trachea, is represented in green. And those are more metabolically active than those seated with a low cell concentration, that is the line in gray. So we compare as positive and negative controls. We compare those results to a native trachea, that is the trachea that it's right away harvested, and that trachea that was only de-epithelized, that trachea that was not seated. So that de-epithelized trachea is in purple, and the native trachea, the metabolic activity of a native trachea is in black. So we have also the histological findings, and we can see that on this slide. We have a native trachea section, and we can appreciate the pseudostratified epithelium and the mucosal glands. That is a normal healthy trachea. So let's remember, as I mentioned briefly during the presentation of our first paper related to re-epithelization and de-epithelization, that we are working with pig's trachea. Pig's trachea are pretty similar to human tracheas, and that's why we are focusing on the porcine animal model. So native trachea is in A. In B, that's a de-epithelized trachea. So we see no cell components, and we just see the ECM. So we don't remove the ECM of the epithelial layer. We keep the ECM and we remove all the cell components. So we don't see any nuclei in that area. In C is a re-epithelized trachea with Bs2Bs at a concentration of 1 million cells per centimeter square. So we can see that even though it's not a completely normal native epithelium, we can see that there are several layers, and the cells were able to attach to that ECM product of our de-epithelialization process. So moving forward, we went even deeper and we stained those tracheas and different sections of the trachea with so we focus on evaluating the high, those tracheas seated with high cell concentration. And that's what we are seeing here. There's a repopulation of the luminal side of the trachea, and we see the Hodge staining that let us visualize each cell as it stains their nuclei component. As we see on top, of each slide, of each picture, we can see several blue points, and those are the Bs to Bs attached to the ECM at the luminal side. What we see at the bottom, for example, in B, those are the nuclei of the chondrocytes, okay? We didn't touch almost didn't affect the chondrocytes, and we have the chondrocytes viable, for example, in that picture. So finally, on this slide, we represent the metabolic activity for 7 days after seeing the tracheas with 1 million cells per centimeter square and keeping the trachea inside the bioreactor for 7 days. So on the right, we have a trachea section of the re-epithelized mucosa in which the green area again corresponds to viable epithelial cells. So this is like a picture on top of the mucosas, looking at the cells on top. So those are viable cells. So these results that I've been showing are available when you read the paper that I just showed. So what do we have by now? So our group tissue engineering approach is decellularization and recellularization of long segment tracheas using a bioreactor, our own bioreactor. So we focus on a partial decellularization protocol, which is the de-epithelialization. The ideal cell seating concentration is 1 million cells per centimeter square of Bs to Bs. So what's the next step? So we need to improve our bioreactor design even more to make it ideal for re-epithelization. So it's perfect for de-epithelialization, so we need to make it ideal, maybe perfect, for our re-epithelization. So we also need to create an ideal environment for the cells in terms of physical and chemical cues. So that's our next step. So in January of this year, in 2021, we thought that in silico modeling of bioreactor modifications will allow for a clear understanding of their effect on the hydrodynamic environment within the trachea. So this would provide guidance to accelerate the optimization of re-epithelization protocols for tracheal scaffolds. So in other words, we optimize the bioreactor to enhance trachea re-epithelization. So in this part, I was working together with the engineers, generating computational fluid dynamic simulations in order to get the results that we got. So our engineers run those simulations of the luminal wall shear stress within the bioreactor. The first generation bioreactor had 2 inlets, and the second generation bioreactor had before 2 inlets and then only 1. And this detail affected our results. So on the left, we have wall shear stress contours, and on the right, the plots. So the shear stress was assessed with the double inlet that it's in A, so on the right left, and single inlet that it's B, and with different rotational speeds. So we wanted to know if low or high rotational speeds were ideal for our cell seating protocol. So we see here that very high shear stress is represented in red, and we can get those shear stress mostly at the end of the trachea at 15 RPMs. So we compare luminal wall shear stress at 1.5 mils per minute at 0 RPM, 1, 5, and 15, as we see on the slide. So we generated these images using ANSYS and MATLAB. So we also simulated particle deposition density. So with only the shear stress information, we couldn't make any conclusion. So we moved to particle deposition density. So that would be where the cells would attach. So the cell deposition density contours of the double and the single inlet designs, as we see A and B on the left. And on the right, we have particle deposition density plots at 1.5 mils per minute as the flow rate. And we also compare those with the double, that is in blue, and single, that is in red, inlet designs. So we also compare different rotational speeds, 0, 1, 5, and 15 RPMs. At this point, we have low shear stress and more uniform cell deposition at 1 and 5 RPM with the single inlet bioreactor. So it seems that that's the ideal design for our protocol. So we decided to move that one moving forward. So we run experiments to validate our simulations and determine which rotational speed will be the best. So with just the simulation, we weren't able to make any conclusion because that's what was generated by computational simulations. So we needed to validate those results with experiments. So here we have the average cell deposition density fraction plot along tracheal length on the left and the circumferential position on the right. So remember that we need to evaluate where the cells attach throughout the whole length of the trachea and where the cells attach around the trachea. So inside the trachea, if they attach at the bottom, on the sides, or at the top. Because remember, the trachea is rotating. So the simulation results that we see here are in blue and the experimental ones are in red. So we can see that there are some difference, but when we evaluated that, we run experiments at 0, 1, 5, and 15 RPMs using the single rotating inlet bioreactor. So these are the results only for the single inlet. So we can see here that at 1 RPM, cells are deposited uniformly, circumferentially, and throughout the whole length of the trachea, okay? And we can see here, let's see, in B and in F, okay? We confirm those results when we run experiments with the single inlet bioreactor at 1 and 5 RPM. So what we did, that we quantified the cells after 2 hours and 24 hours in the bioreactor at 1 and 5 RPM. As we can see on this graph, we concluded that sitting at 1 RPM would result in higher number of cells on the trachea. So here we have the data presented as means with standard deviation, and each group was 3 tracheas. And we can see the statistically significant difference between both groups. So we are going to move forward with the 1 RPM. So here we have the results for long-term re-epithelialization experiments at 1 RPM in the single rotating inlet bioreactor. So before we did 2 hours and 24 hours, and we went even longer, 5 hours. Inlet, middle, and outlet sections is pretty, it seems pretty uniform. And but still, the cells are in a monolayer, in a flat monolayer. So we can see as well the live death staining of the inlet and outlet sections in D and E, and the metabolic cell activity over time compared to the de-epithelized tracheal graph, as we can see on the on F, and the number of cells per field in G. So it looks pretty consistent in the inlet, middle, and outlet of the trachea. So what do we have now? An enhanced bioreactor for de-epithelialization and re-epithelization, and clear physical conditions required. That is shear stress with a rotation of 1 RPM, so it would be a really low shear stress, and flow rate of 1.5 mils per minute. So what's next? We need to achieve differentiation in the bioreactor and use clinically relevant cells. That would be the human-induced pluripotent stem cells. So let's go into that area. So the airway epithelium needs to be exposed to air in order to differentiate. Bs2Bs cannot be used in this part of the project because Bs2Bs are not meant to be differentiated even when exposed to air. So we have to think about other kind of cells. So that differentiation, that in vitro, we can make possible this differentiation starting with a monolayer of cells, and then it would become a pseudostratified mature epithelium after some time of exposure to air, as we see on the right, labeled as B. So we can see on the slide a schematic representation of submerged and air-liquid interface culture of primary human bronchial epithelial cells. These cells are growing using porous culture inserts, and we call them transwells. So we can remove the top, and then it would be two different areas. So the bottom of the well, and at the top we will have the transwell. So at the beginnings, the cells are submerged, as we see in A, and then when they are fully confluent, they are all them in contact between them, and are in contact with the media that we see also at the bottom and at the top. When we move forward and we see that the cells are ready to be differentiated, we expose the cells to air, and they will be in contact with media only at the bottom. So we had to recreate this system, this in vitro system that is well established, using our bioreactor system, because when it's already, the cells are already seated, we had to differentiate the cells inside the trachea. We cannot move back to in vitro. So we run experiments using native trachea. So it will take a lot of time, a lot of resources and a lot of trial and error to test the ALI system in the bioreactor with cells. So what we thought, okay, let's work with native tracheas. So when the epithelium is no longer exposed to air, for example, if I submerge the trachea in media, the epithelium will no longer be ciliated, pseudostratified. So it will differentiate to a monolayer or a flat, partially pseudostratified epithelium. Okay, so it wouldn't be functional anymore. So before we use the system that we have on the left, that was the system that we created for re-epithelization. We just needed, because that's completely submerged, as we see at the bottom left, it's complete in red, the inside of the trachea, it's completely, the trachea is completely filled with media. Then when we created that system, the differentiation in that moment was not considered. But after we achieved the results that we got, well, then we had to modify that system and added humidified air into the luminal circuit. And that's what we see on the right. We see the Erlenmeyer with air and water, and we are pumping, in that case, air to the system. Okay, so one pump will pump air and another one will pump media, and then when combine both components, there will be bubbles surrounded by media flowing into the lumen. So on top of that, we had to tilt the bioreactor, as we can see at the bottom right, so the cells will be equally exposed to air and media while the bioreactor is rotating the trachea. And these were the results. So tracheas in liquid-covered culture, or LCC, or submerged culture, that we can see on the top, showed a monolayer of somewhat flat or cuboidal cells. Tracheas in air-liquid interface culture, or ALI system that we created, that we can see the results at the bottom, showed ciliated pseudostratified epithelium. So probably functional epithelium. So we were able to create an air-liquid environment within our bioreactor system. So the next step would be find the right cells to develop engineered tracheas, because we were working with native tracheas. So we consider in that moment using primary cells, but because of the issues that were surrounding the primary cells, we consider using human pluripotent stem cells. So stem cells are those with the potential to develop into many different types of cells in the body. So they serve as a repair system for the body normally. So we'll focus on our attention on induced pluripotent stem cell. And this is a subject that it's really explore in the last 5 to 10 years. So briefly, we'll see what that means. So we have an schematic view of human pluripotent stem cell induction and airway cell differentiation. This image was taken from the paper that I showed before, which is not ours, but which is really relevant to what we are going to discuss briefly after this slide. So there are two ways to obtain human pluripotent stem cells, or HPSCs. The first way involves isolated inner cell mass from blastocyst embryo to obtain embryonic stem cells. Another way is by using human somatic cells. These somatic cells are exposed to transcription factors, as we can see on the slide, to induce induced pluripotent stem cells. ESM, so embryonic stem cells, and pluripotent stem cells are together referred as human pluripotent stem cells. So from here, the human pluripotent stem cells can be differentiated into other cell types, among which are a variety of cell types that can be found in the trachea and lungs. So it would be alveolar type 2 cells, ciliated cells, goblet cells, and all the different cell types that we can see on the airway epithelium. So one of our group members, Dr. Varma, mastered the in vitro culture of airway progenitors derived from human-induced pluripotent stem cells following the Cotton protocol from, this is a group that it's located in Boston, US. So Ratna was able to differentiate the airway progenitors into differentiated epithelia in vitro. So we would use those airway progenitors after 42 hours of in vitro culture, seed them onto the de-epithelized trachea in the bioreactor system that we created, and differentiate them using the ALI bioreactor system that we already created with native tracheas. So we accomplished that goal up to 7 and 14 days in the bioreactor. We weren't able to complete all the tracheas needed for each group because of the pandemic that started last year. So, but with those preliminary results, we observe partial differentiation and early stages of ciliation of that epithelium that was created. So it was not a monolayer, they were not flat, they were not cuboidal, they were pseudostratified, almost creating cilia by day 14. As I said, the data is not shown and not published yet, but these are promising results to continue developing engineered tracheas in the lab using clinically relevant cells as are the iPS cells. So what do we have by now? The bioreactor ALI system. We also accomplished the Re-Epithelization with human-induced pluripotent stem cells, and we were able to get human-induced pluripotent stem cell differentiation within our bioreactor. So what's next? Differentiation to fully mature epithelium and transplantation of engineered tracheal graft onto an animal model. And that's the stage that our project is. So let's finalize our discussion today with this quote from Dr. Lanza, a recognized American scientist working in the field. In fact, when you combine stem cell technology with the technology known as tissue engineering, you can actually grow up entire organs. So as you suggest, sometime in the future, you get in an automobile accident and lose a kidney. Would simply take a few skin cells and grow you up a new kidney. In fact, this has already been done. Now is the future. We will create customized tissue engineered organs with autologous pluripotent stem cells. As I said, we are already working on this. Thank you very much. It was a pleasure to share this presentation, and right now I'm happy to answer any question.
Thank you so much, Alba. I would like to ask you, well, you already stopped sharing your screen, and now we can start the question session. So let's start with questions from the participants that were sent via chat. So the first one is from Pedro. He is asking, what is the purpose of the rotation in your bioreactor system?
Okay, this is a very good question because I didn't mention that. So the rotation would make possible that the cells are not in contact. For example, if the trachea is not rotating, the cells would flow, and most of the cells would attach to the bottom of the trachea. So that's not the goal. The goal is that the trachea is fully repopulated, but not only at the bottom, but throughout the whole circumference of the trachea. So that's only possible rotating the trachea during the process. So you are injecting the cells and the trachea is rotating, so the cells would be in contact with the mucosa of the entire trachea. So that's why we have created this system with rotation. So this is not our our novel approach that was created by our group. There are different groups around the world that are working with the trachea, and this is an approach that is pretty common in this field. The other, the question would apply as well to the de-epithelialization. So why you are exposing the trachea to the detergents rotating, and it's the same principles. So you are rotating the trachea, so the flow of the detergents are acting in the whole length of the trachea and circumferentially as well, and not just exposing one section of the trachea. So you're exposing the whole mucosa. I hope this answered the question.
Yeah, somehow the second question is related to the first one, but it's a more like a physical question based on, so you showed that there's in Lee's paper that there's a difference between, so the flow is one way, so it goes from the inlet to the outlet, so it goes in this direction, right?
Yes.
So this person is asking, how do you explain the difference between different concentration between inlet and outlet compared to the middle, right? In the case of higher cell concentration, I think it's related to, could you explain how the simulation is related to answering this question, this matter?
Yeah, so if we think about the process, the cells would be, as Fabio mentioned, injected into the inlet, okay? So it would be more cells, the mucosa at the inlet and middle will be exposed to more cells okay? The number of cells is higher than the ones that reach the end of the trachea, the end so it would be the outlet of the trachea. So the cells are attaching while flowing, and we need to remember that flow that we are using, it's really, really low flow. It's 1.5 mils per minute. So it's a really, really low flow. And at the beginning, it's a high density mixture of cells and media, but it's a really dense composition. So as mentioned before, we can see, as Fabio said, in the simulations as well, that there are more particles attached at the bottom, at the inlet, then at the middle, and then at the outlet. Even though we thought that difference was not a big issue, since after some cells are attached, they will expand themselves, and then as soon as they have some kind of cell interaction, they will start the proliferation.
Thank you, Alba. And now, Professor Tatiana, would you mind continuing with the question?
Yes, sure. It's a pleasure. We have another question from the audience, from Clark Louise. In your view, what are the expectations of the electrospinning technique in tissue engineering?
Well, this is a very good question, and even though it's not my specific area of specialization, as I mentioned, there are a lot of approaches, different protocols, and each protocol have their pros and cons. Okay, we think that our approach is a better option because we'll use non-viable organs to make them scaffolds, and then we can use those non-viable organs from deceased donors to create new organs. So we wouldn't need like high really specialized technology in order to get these results.
Okay, thank you. And I have one question. How do you, it's a curiosity, how do you estimate the ideal distance between the cells when you do the cell culture, considering that this distance between two cells interferes in cell proliferation and maybe in your Re-Epithelization process? How do you estimate? You count the cells and then
it's a good question, and it's really focused on the in vitro. That's where we can see and quantify what's going on during the culture, because it's hard to have an idea of what's happening inside the trachea. So in vitro, we count the cells during each passage. So in those let's focus on the creation of the airway progenitor. So that protocol of 42 days in vitro. So we count each in passaging the cells. So we passage 2 or 3 times, and in each passage would be counting how many cells we have. And then we can estimate how many cells we will have at the end, and that it needs to be, for a long segment trachea of 5 centimeters, it needs to be 20 million cells. That would be 1 million cells per centimeter square. If we sit less than that, then we won't have the results that we need. So if we, as I said, if we sit less than that, for example, 750,000 cells or 500,000 cells, then the repopulation would not be enough. There won't be enough cell attachment to create that proliferation and then the differentiation. If the cells are not attached, then the cells won't initiate the differentiation process. So what was my estimation of maybe the time? It's, as I said, that cotton protocol that Radna mastered during her PhD project. It's pretty straightforward. It can be followed step by step, and you will be able to create during the 42 days, but after a really long time experimenting with those cells, you will be able to create the cells that you need, and then you will be able to see the trachea. So it's a pretty long process, even though the de-epithelialization was created really short, because like fully decellularization would take several weeks, even a month, to fully decellularize the trachea, even the cartilage. And Fabio was able to keep it short in less than 6 hours, and that saves time. But at the same time, we'll need to be creating the airway progenitors with the human-induced pluripotent stem cell protocol, and then we can match the timings. And then if you add up all the periods, it would be 42 plus 3 plus 14. So it would be around 2 to 3 months to create these engineered tracheas. After that time, then you will be able to transplant the trachea.
Wonderful, wonderful. Congratulations, Alba, about this wonderful work of that you have been developing and conducting, and also congratulations to your team. I know that Fabio and you worked together, so congratulations about this. And I have one more question. May I go ahead, Fabio? Is it okay for you? I have one question about the ethical issues about working on projects like yours. What are the main ethical, not problems, we could not say problems, but barriers that you find in your work studying or conducting this kind of study?
Yeah, that's a good question, because that's something that we need to deal with, and that's beside the lab work or the work with the bioreactor and all those things. Of course, during the computational simulations, there's no issue, so we can run as many simulations as we want. But as soon as we start, for example, working with animals, we need to work together with a group of vets, with a group of veterinarians and technicians, in order to make those surgeries and those harvesting protocols in a humane way. So in our case, our pigs are treated like strictly as patients. Is even if you are working with humans, it's exactly the same. The ORs are the same. The anesthesia is the same. Everything is the same, and our protocols needs to be approved by a board and an ethical board in order to move forward. That's when we focus on that animal model. So even in order to harvest the trachea, we need permission. That's what I mean. When we want to move forward, working with human tracheas, that's something that it's possible as well. And we've been working with human tracheas as well, because we were in another side of the project, we were using human tracheal epithelial cells. So those epithelial cells harvested from a segment of a human trachea. We had a permission from the ethical board of the hospital that we were getting the tracheas. So the donors, in this case, were deceased donors, the family of the donors, and if the donor had consent to donate their organs, either for other people or for research, we were harvesting, we were getting those tracheas with permission of the board. That's the other thing. And when we were harvesting the tracheal cells from the human tracheas, that have to be specified in a protocol that we sent before starting the project. In terms of using different kind of cells, embryonic stem cells are the ones that are surrounded by more ethical issues, because you have to use, in order to get those, you have to use embryos. So you have to use eggs that were already, that the fecundation of the process of the spermatozoid and the egg were already, and an embryo was being developing for several days. It would be 5 to 7 days. In that moment, those embryos will be used to get those cells, those stem cells. That was at the beginning. But then more research was done in the field, and that's when we have the induced pluripotent stem cells. So these cells would be, we will have those cells when we work with somatic cells. That's why at the end of the presentation, the quotation of Dr. Lanza mentioned that even getting skin cells or bone marrow cells or fat cells would be, those cells would be the source of these differentiation protocols. So starting with those cells from the recipient, we'll go through the whole process, and we will be able to get even, for example, cells for smooth muscle, cardiac cells, lung cells, any cell that you need in the body. Of course, in order to differentiate those stem cells that were harvested from the patient that needs the organ, needs to be differentiated with specific protocols. Some protocols are well established, and some other protocols, for example, the heart team in the field has developed a really detailed protocol, and they are able even to inject those cells in patients that has had a myocardial infarction. So those protocols are maybe a little bit ahead of ours, and they are already being used in the clinical side. So when you work with embryonic stem cells, there's a lot of ethical issues, because the couple that donated those cells in order to get a fertilized egg would have to consent to give that embryo to research. And of course, there are a lot of also religious groups that are not, that do not, and not only religious group, but around the world, there are a lot of researchers that are not, how do you say, are not, do not agree with that process, because that's a lie that it's being used to research, and then it would be used to create an organ. So it's a life to another life. So there's a lot of like a lot of room for discussion in that specific area in terms of the embryonic stem cells.
Okay, thank you very much, Alba. Thank you.
Thank you both for the invitation, and thank you to all the participants, and thank you for the questions. They were a very good question, and like we had a really good discussion.
Okay, thank you, Alba. I was wondering here how amazing your work is, because we belong to this biomedical engineering group, right? And it's amazing how many skills you have, because as a medical doctor, you had to go through another field, which is the engineering field. So we had to learn about basic engineering, like designing a new bioreactor and etc. And then you had to learn about how the shear stress affects the lumen of the trachea, and how the rotation affects, right, the cells to attach to the lumen, and how does. So it's we're talking about two like different areas that are tightly connected, right? And this is really amazing, right? And you didn't mention, but you also participated in the surgeries to transplant some some preliminary tests to transplant the tracheas, right? So you were there. So you started in the lab in basic research, passaging cells, and then you went to the bioreactor designing, and then you after that you went to the OR operating room and transplanted.
Translational science, wonderful.
Yeah, the excitement is where you can see how you can translate basic science and bioengineering approaches into a possible solution for patients waiting for transplant. So it's not too far from, well, it's really far, but it's also related to the work that I was doing in Venezuela with kidney transplantations. So when you have patients that are in need, like it's critical, it's life-death issue for them to get new organs, and then you are able to see that in a lot of places in the world are working towards helping these patients to get new organs, then it's like I feel that there's hope. Like there are a lot of things going on, like really bad in the world, but then you can have like you can work towards the solution and not only focus on the problem. So yeah, it was really, I was really excited. At the beginning, I was really lost, but then you make the whole big picture in your head after working for several months and maybe years, and then you can have the idea of what you want to do and how you want to do it.
Congratulations.
And as I mentioned, sorry, Fabio, just one last thing. As I mentioned, I didn't work alone. There are a lot of people behind these projects. As I said, there are several publications, and all of them have several authors. And of course, the principal investigators have been working in these projects for a really long time. And that's why it's possible to have really good results, really promising results when we are focusing on the preliminary part, and that's when the excitement starts.
Congratulations on your work, Alba. It was unfortunately our time is short, but I'd like to thank you again for your availability and for the excellent lecture on tracheal tissue engineering. I would like to tell the participants that the event continues with lectures or presentation sessions. I think it's today, right, Tatiana, elevator pitch competition, which will be very exciting, the roundtable and mini course. So thank you again, Alba. It was you are more than welcome to visit our university here in Brazil, and let you know that you're a very fan of your work, and thank you for everything.
Thank you.
Thank you, Alba. Thank you.
Nice to meet you, Tatiana, and nice to meet you all. Thank you. It was a wonderful time.
Okay,
Bye-bye.
Congratulations. Bye-bye. Bye.